Good morning. Can you hear? So there is SSC lecture now. Last time we have finished two lectures. In the last lecture, we had seen the different zones in the water ecosystem, which involves littoral zone, limnatic zone, profundal zone, fresh waters, including rivers and streams, marine waters, including estuaries, mangrove ecosystem, abyssal water column or deep sea, and hydrothermal vents, including coral reef, which are present in the hydrothermal vents. I think hydrothermal vents was remaining. So these hydrothermal vents, these are nothing but small fissures or openings in the floor of the ocean, and they release hot mineral rich water. So this thermal indicates temperature, hydro means in water. So these vents, these are nothing but openings in the floor of the ocean, and they release hot mineral rich water. This temperature of this hot water reaches up to 750 degrees Fahrenheit. There are two types of hydrothermal vents. The vents which are very hot, they are known as black smokers. And in this case, dark smoke, which is composed of iron and sulfide, it comes out. And a bit cooler ones are white smokers. So it contains less uh, temperature as compared to the black smokers, and they release compounds like barium, calcium, silicon, etc. So hydrothermal vents, they are of two types. The vents which are very hot, they are known as black smokers, and the vents which are comparatively cool are known as white smokers. The black smokers, they are composed mainly of iron and sulfide, while white smokers are they release compounds like barium, calcium, silicon, etc. These hydrothermal vents, they are found in the ocean floor, so it is very obvious that they may be found in Pacific, Atlantic oceans at a depth of 2,100 meters, almost 7,000 feet below the surface of water. The ecosystem of the hydrothermal vent, it contains only specific type of adapted life forms, means only specific type of microorganisms. These organisms, they mainly include some species of thermophilic bacteria, tube worms, mussels, crabs, lobsters, octopus, and even fish. Some animals are also found in the hydrothermal vent ecosystem. Now these hydrothermal vents, they are almost 7,000 feet down. So it is very, very obvious that there will be no light 7,000 feet below the surface of the water. So photosynthesis will not be there, or photosynthetic activity will not be visible by algae which are present. So normally the production of food material or nutrition will be by chemosynthesis. So photosynthesis and chemosynthesis. Photosynthesis will be in presence of light. Chemosynthesis will be presence of in presence of certain chemical compounds. So this process, it requires specifically archaea type of organisms which can convert chemical energy into some usable form of energy. And all these chemosynthetic bacteria, organisms, or algae, or animals, they form a special food web in the hydrothermal vent. So just like the food web on the in the ponds, the food web on the land surface, okay, which involves producers, primary consumers, secondary consumers, tertiary consumers, and decomposers. So similar type of food web, but here there will be, now normally on the land, producers are photosynthetic plants. Okay, so they produce plants. Then there are primary consumers, which are usually herbivorous animals. Then on these herbivorous animals like rabbits, deer, secondary consumers will be there, and slightly larger than them. And then there will be tertiary consumers, which involves lion, tigers, vultures, etc. And when these die, even producers and consumers, when they die, they will be decomposed by the decomposers. Detrimental activity will be carried out by decomposers. So this is the food web which is present on the land. Similar type of food web is also present in the hydrothermal vents. वेंट म्हणजे काय, असे खाच असतात, ओपनिंग्स असतात. आणि कुठे असतात ते, ओशन खाली, वरती पाणी असतं ओशनच्या, आणि खाली जिथे ओशनची जमीन असेल त्याच्यामध्ये हे वेंट्स असतात किंवा हे ओपनिंग्स असतात. आणि त्याच्यातनं गरम पाणी बाहेर निघत असतं, सोबत त्या जमिनीत असलेले काही मिनरल्स सुद्धा बाहेर पडत असतात. सो इफ वी कन्सिडर दी ब्लॅक स्मोकर्स, टेम्परेचर जास्त असतं ते हॉट वॉटरचं, आणि कूल स्मोकर्स और व्हाईट स्मोकर्समध्ये टेम्परेचर कमी असतं. हॉट smokers or black smokers, they release mainly iron and sulfide, while white smokers, they release barium, calcium, silicon, etc. So they are found in Pacific and Atlantic Ocean, and they consist of specifically adapted forms of life. No photosynthetic activity takes place in the hydrothermal vents because there are no chances of sunlight reaching to a depth of 7,000 feet or 2,100 meters below the surface of water. Now, coral reefs. These coral reefs, they are composed of thin plates of calcium carbonate, which are secreted by soft-bodied animals like coral polyps. So these polyps keep on liberating calcium carbonate or secreting calcium carbonate, and these layers of calcium carbonate go over each other and form these corals. So these coral polyps, they are found in symbiotic association with zooxanthellae, and they give typical colors to the coral reefs. So zooxanthellae will be the animals, and they take in carbon dioxide through photosynthesis, and they give off oxygen and other nutrients which are used by host polyps. So coral polyps and zooxanthellae, they are found in association. What does zooxanthellae do? They carry out the process of photosynthesis. They liberate oxygen and nutrients, while the coral polyps, they consume that oxygen and nutrients. Coral reefs are very diverse ecosystems on the earth, which include corals, sponges, crustaceans, mollusks, fish, sea turtles, sharks, dolphins, etc. You can see they start from coral reefs, then they go to sponges, then they go to crustaceans, mollusks. ते साईज त्याची मोठी मोठी होत जाते. Fish, sea turtles, sharks, dolphins, etc. Is this clear? Coral reef and hydrothermal vents. Microflora of water we have finished. Now we'll twist over to the bacterial assessment and potability of water. So, which water is safe for drinking? You will say filtered water is safe for drinking. RO water is safe for drinking. But as a microbiologist, it is necessary to know which type of water is actually free of pathogenic organisms, and what is the indicator will tell which tells you that this water is safe for drinking. So as a microbiologist, you should have knowledge about this. Now, the indicator bacteria will give you an indication whether the water is good for drinking or not. Now, these include process indicators, means when you treat the water, after treating the water, uh, if these organisms are present, they will tell you that the water is not safe for consumption. So, if these organisms are not present, these are the indicator organisms. You know, in a vehicle, what does the indicator do? It will tell you whether to go. The person who is coming behind or the vehicle coming behind will come to know whether to go right or left because of the indicator lights. Same way, this indicator bacteria, they will tell you whether the water is safe for drinking or not. Now, these indicators are of different types. These bacteria, if they are present, they will tell you that this process is not properly done. So, process indicators are organisms which will determine the efficiency of the process or efficacy of the process. These include the heterotrophic bacteria, the coliforms, and usually after chlorine disinfection. Okay, so if water is to be consumed, even if it is river water, bore water, any type of water, we carry out chlorine disinfection. So after this disinfection, this bacteria, heterotrophic bacteria or E. coli, their number should be reduced considerably and they should not be present in large numbers. So if they are present in a count more than a specific level, that indicates that the processes the process indicator organisms are present, and so the process is not done properly, so it should be repeated. That is, chlorine disinfection should be repeated. The other one is the fecal indicator. So these are the organisms which indicate the presence of fecal contamination, whether fecal bacteria are present or not. So if these organisms are present in water, they will tell you that this water has been contaminated with fecal matter. Okay, waste, human waste, animal waste, त्याच्याने ते कॉन्टॅमिनेट झालेलं आहे. तर असे कोणते बॅक्टेरिया त्याच्यात पाण्यात प्रेझेंट असले तर आपण सांगू शकतो की दे आर that water is not safe for consumption. They include the thermotolerant coliforms or E. coli. The coliform organisms. What is the meaning of coliform? Coli means intestine. So the bacteria which are present in intestine, they are known as coliform. Thermotolerant means they can tolerate high temperature. So such high temperature tolerating intestinal bacteria, if they are present in water, or certain E. coli, which is also an intestinal bacteria or coliform bacterium, if it is present in a definite number or if it is present beyond a definite number in the water, then you can say that that is fecal contamination of water and that water is not safe for drinking. Presence of such type of bacteria indicates that pathogens may be present. Now it is not possible to check each and every organism ना, आपण काही प्रत्येक ऑर्गॅनिझम पॅथोजन खूप आहेत. साल्मोनेला टायफी, शिगेला डिसेंट्री, विब्रिओ कॉलरी, हे सगळे बॅक्टेरिया पॅथोजन्स आहेत, इंटेस्टिनल बॅक्टेरिया आहेत आणि ते भयंकर आजार कॉज करतात. डायरिया, डिसेंट्री सारखे ऑर्गॅनिझम्स डिझिजेस कॉज करतात. ते पाण्यामध्ये प्रत्येकाला शोधणं फार कठीण आहे. त्याच्यामुळे आपण काय करतो की त्याच्या बरोबर असणारे इंटेस्टिनल बॅक्टेरिया यांचा काउंट इतक्यापेक्षा जास्त असेल तर हे बॅक्टेरिया पण प्रेझेंट असण्याची शक्यता आहे. So presence of thermotolerant coliforms or E. coli indicates that there may be the presence of other pathogens which are thermotolerant coliforms like Salmonella shigella, Salmonella typhi, Shigella dysentery, Vibrio cholerae, etc. So that water is not safe for drinking. So please see that how these indicator bacteria are telling you that water is safe for drinking or not safe for drinking. If these thermotolerant coliforms or E. coli are in very negligible number, खूप मोजके 10 पेक्षाही कमी जर तुम्हाला काउंट करता येतात, ते काउंटिंग प्रोसिजर आपण बघणारच आहे नंतर. So if you find them to be in a negligible or very less count, then you can say that that water is not contaminated with other pathogens, and there are no chances or very less chances of presence of this pathogens. So the water is safe for drinking. So process indicator will tell you, these bacteria or these organisms will tell you whether the disinfection procedure which you have carried out is done properly or not. So, in spite of doing disinfection by chlorination, if certain bacteria, heterotrophic bacteria and coliforms, if they are present in large number, this means chlorination has not been done properly. The efficacy of chlorination is not okay, not fine, not up to the mark, and that is why uh, that water needs to be processed once again or treated once again. The fecal indicator bacteria or organisms will tell you whether the water is fecally contaminated or not. These bacteria or these organisms include thermotolerant coliforms or E. coli. If their number is very high in the water, then you can say that this water is not safe for drinking, because there are chances of, if these organisms are present, means there may be presence of other pathogens which may be very very harmful for the human beings, and that is why this water needs to be treated or the contamination needs to be taken care of. Next are index and model organisms. So what is an index? Indicator आणि index organisms याच्यामध्ये फरक आहे. Definitions are asked. Remember, in examination, indicator organismची definition वेगळी असेल, index and model organismची definition वेगळी असेल. So a group or species which indicates the presence of pathogen and behavior, such as E. coli is an index for salmonella. So if E. coli is present, salmonella 99% it may be present. And FRNA coliphages as models of human enteric viruses. So if bacteria आपल्याला consider bacterial pathogens consider करायचे असतील, तर असे organisms जे indicate करतात की E. coli is an index organism, but it is an index of what? It is an index for presence of Salmonella typhi, which is responsible for enteric fever or typhoid. Now, FRNA coliphages, if they are present, coliphages means what? The viruses which eat bacteria. There are three types of viruses: animal viruses, plants viruses, and bacterial viruses. Animal viruses, they will infect animal cells, including human cells, like coronavirus is an animal virus. SARS virus is an animal virus. So Nipah virus is a animal virus. तर हे जे व्हायरसेस आहेत ते ह्युमन सेल्सला अटॅक करतील, ॲनिमल सेल्सला अटॅक करतील. प्लांट व्हायरसेस असतील ते प्लांट सेल्सला अटॅक करतील. आणि तिसरे जे आहेत ते बॅक्टेरियल व्हायरसेस किंवा बॅक्टेरिओफाजेस आपण म्हणतो त्याला, तर ते बॅक्टेरियल सेल्सला अटॅक करतील. आणि कोलिफाजेस म्हणजे काय? तर जे कोलिफॉर्म ऑर्गॅनिझम्सला इन्फेक्ट करतील. कोलिफॉर्म ऑर्गॅनिझम्स म्हणजे काय? तर those found in the colon or the intestine. So coliform organisms, if these phages which are infecting coliform organisms are present in water, it is an index that other human enteric viruses may also be present, like rotavirus, which causes severe diarrhea, Coxsackie virus, and other viruses. So those viruses are definitely present if FRNA coliphages are present in water. So these are the index and model organism. Index organism is with respect to another organism which is a pathogen. So E. coli is an index for Salmonella. FRNA coliphages are index for or models of human enteric viruses. Now, what is the criteria of indicator organisms? Whom can you call an indicator organism? Or which organism can be referred to as indicator organisms? So, the organism should be present whenever enteric pathogens are present. So, they should always be present along with enteric pathogens. It should be useful for all types of water, then only you can call it as indicator organisms. It doesn't mean that it is good for fresh water and it is not good for other types of water or marine water. No, an indicator organism should be useful for all types of water. फक्त river water नाही तर well water साठी, stream water साठी, bore water साठी, for all types of water, this organism should show the same type of results, then only it can be called as an indicator organism. It should have a long survival time than the hardiest enteric pathogen. So, of all the enteric pathogens, सगळ्या enteric pathogens पेक्षा त्याचं सर्वायवल टाइम खूप लॉंग असलं पाहिजे. म्हणजे डिसइन्फेक्शन केल्याबरोबर मरून जातील काही एंटेरिक पॅथोजन्स, पण तरी पण हा जिवंत राहिला पाहिजे. अशा स्ट्रॉंग ऑर्गॅनिझमला तुम्ही इंडिकेटर ऑर्गॅनिझम म्हणून कन्सिडर करू शकता. सो इट शुड नॉट ग्रो इन वॉटर. वॉटर त्याचं न्यूट्रिएंट नसायला पाहिजे. इट शुड नॉट, वॉटर शुड नॉट ऍक्ट ॲज अ न्यूट्रिएंट. Then it should be found in warm-blooded animals, intestines of warm-blooded animals like human beings, other mammals. This organism should be found in their intestine. So E. coli is the best indicator organisms. Which are the 5 conditions for any organism to be called as an indicator organism? It should be present where enteric pathogens are present. It should be useful for all types of water. It should have longer survival time than the most strongest enteric pathogen. It should not grow in water, and water should not be a nutrient, and this organism should be found in warm-blooded intestines. Let us consider the first enteric organism, Escherichia coli or E. coli. This Escherichia coli is a rod-shaped organism found in the gastrointestinal tract, that is intestine, and feces, the waste material of warm-blooded animals. Human beings, they are warm-blooded animals. All mammals, they will be warm-blooded animals. The harmless strains are the part of the normal flora of the intestine or gut. So, E. coli is the normal flora of your intestine. It is always present in your intestine. They provide vitamin K to us, and they also prevent other pathogenic organisms to survive in the intestine. Presence of E. coli is in the intestine is as good as the presence of सिक्युरिटी गार्ड ऑन द ग्रेट. सिक्युरिटी गार्ड जसा दुसऱ्या कोणालाही किंवा आतंकवादीला किंवा कुठलाही नक्षलवादीला आतमध्ये येऊ देणार नाही. प्राईम मिनिस्टरच्या कॅबिनच्या बाहेर सिक्युरिटी गार्ड्स असतात, तर ते आतमध्ये येऊ देणार नाही नको त्या लोकांना. तसं ई कोलाय सुद्धा पॅथोजेनिक ऑर्गॅनिझम्सला, डिझीज कॉझिंग ऑर्गॅनिझम्सला इंटेस्टाईनमध्ये येऊ देत नाही. एवढंच नाही तर इट ऑल्सो प्रोव्हाइड्स विटॅमिन के to human beings. It is the only member of the coliform group which is found in the intestine, and it will indicate, because it is present in human intestine, it will be present in human waste, human fecal matter, and thereby presence of E. coli in water will indicate fecal pollution of water or fecal contamination of water. What are polyforms or E. coli? They are aerobic and facultatively anaerobic. Means they can even grow in absence of oxygen or little quantity of oxygen. जर खूपच कमी ऑक्सिजन असेल किंवा एनारोबिक कंडिशन्स असतील तर तिथेही तो ग्रो होऊ शकेल. एरोबिक आहेच, ऑक्सिजनमध्ये ग्रो होऊ शकेल, ऑक्सिजनच्या ॲब्सन्समध्ये मरून जाणार नाही. ग्रॅम नेगेटिव्ह नॉन स्पोर फॉर्मिंग रॉड. It's gram negative in nature. It doesn't form spores. It can ferment lactose and produce acid and gas within 48 hours at 35 degrees Celsius. So how will you define polyforms? You are going to learn this polyform definition perfectly. लवकरच तुमचं कॉलेज सुरू होणार आहे, मी हे डेफिनेशन्स तुम्हाला विचारेन. सो व्हॉट आर पोलीफॉर्म्स? Underline it. When I send you the notes, underline it and learn it. Coliforms are aerobic, facultatively anaerobic, gram negative, non-spore forming rods which ferment lactose and produce acid and gas within 48 hours at 35 degrees Celsius. Clear? So E. coli is a member of fecal coliform group and it cannot break down urease. Sorry, it can break down lactose, but it cannot break down urease. So urea. So it doesn't contains enzyme urease. युरियाला तो ब्रेकडाउन करू शकत नाही. So urease is absent. This means that it cannot produce the enzyme urease. These E. coli are found in fresh water, and if you grow them on specific media, you can get, like if you take 0.45 micron filter and placed it in empty EC medium, you get yellow or yellow-brown colonies after incubation at 35 degrees for 24 hours approximately. If you add urea, then addition of urea substrate confirms that the colonies are E. coli because it cannot break down urease. This bacteria is preferred indicator for freshwater recreation and its presence provides indirect or direct evidence of fecal contamination of water. So let us stop here and continue it in the next lecture. तुम्हाला परत दुसरं कुठला तरी लेक्चर असेल. आपलं इथपर्यंत झालेलं आहे युरिएज पर्यंत. So we'll stop here and continue it next Wednesday. And Saturday your lecture will be there at 2 p.m. So join for your lecture at 2 p.m. on Saturday. We will be continuing microscopy on Saturday and Monday. And Wednesday there will be SSC lecture.